Okay, good morning. Today we're going to talk about GWAS, which is to say genome-wide association studies. Okay, the idea of this is try to locate genes that are involved in disease. And we're going to use a slightly unusual method called single nucleotide polymorphisms to do that. Okay, so the idea is that single nucleotide polymorphisms are locations in the genome where there's genetic variation, where people differ one from the other in terms of what actual nucleotides they have at that location. And the important thing is we know where they are, and therefore the idea is going to be we're going to try to correlate disease susceptibility to the locations of particular SNPs. Okay, so a single nucleotide polymorphism is basically one of the millions of sites in our genome where there is variation from one person to another. So for a particular SNP, a person might have a genotype AA, AT, or TT. So even though it's not a gene, you certainly have a genotype at each of these millions of loci. So you can see these as a genetic marker in a sense that we can use in essentially a linkage approach to try to locate the disease gene that we're interested in. Okay, so this exemplifies what a SNP is, and we'll get back to this slide a little bit later. The idea is, for example, SNP1 has either an A or a G at that location. SNP2 has either a C or a T, SNP3 an A or a C, etc. So there's variation across the genome at specific locations, and people differ in those things. And we're going to use that to try to locate not the disease gene itself, that we can't do yet, but actually just pinpoint the location in the genome where that disease susceptibility is found. Okay, so we're going to use this enormously high-tech approach, which is called microarray. This is only one possible microarray platform. But the idea of these microarrays is we can actually genotype hundreds of thousands of SNPs all at the same time on one tiny little piece of silicon, which is referred to as a chip, although it's not really a chip. Okay, so the smallest one, and this is the Illumina platform, there are others. The smallest one, in the sense of how many oligos it has about 750,000, and you can actually genotype 12 different people using that one array. And the largest one has 4 million oligo probes, which is kind of astonishing. Okay, so the way this approach works is using these teeny little beads, which are very, very small, and they're embedded in a silicon wafer. And each of those beads is coated with specific oligonucleotides, whose job it is to detect particular SNPs at particular loci. So each probe is designed to determine the genotype at one of those millions and millions of SNPs. Okay, so the oligonucleotide matches the DNA immediately adjacent to the SNP variant nucleotide, and that's kind of key to this particular type of microarray. So what we're going to do is take fragmented DNA from a person, hybridize it to the whole chip, so you've got millions of hybridizations going on all at once. And the difference in this particular approach is that the DNA oligo will be extended by one nucleotide, and that will be labeled with a dye so we can see it later. And the color will tell us the genotype of that SNP. Okay, so what this looks like, you have fragmented DNA from a person, and the DNA is hybridized to these millions of beads all at once. And the key here is that the oligo is set up so it's one nucleotide offset from the nucleotide that you're trying to see. So the person's DNA here has a T, and we expect that it will then match with an A, or be matched with an A by DNA polymerase. So what happens here is we've got our T, DNA polymerase will be added along with fluorescent nucleotides, which are labeled with colored compounds so that they can be detected later. So it's a single nucleotide enzymatic extension of the oligo probe to the person's DNA to identify that particular nucleotide. Okay, so what this looks like after hybridization, sorry this slide's kind of blurry, this person has a T in their DNA, it will match with the oligo, then extension will cause an A to appear there, and it's a fluorescent A, so it can be then detected later by imaging the whole bead array. This other person this other SNP has a G in the person's DNA that will be extended by including a C on the oligo. And you can be heterozygous, and this person has a G and a G and a T in their template. And those can be distinguished by the fluorescence that's obtained from that particular bead after hybridization and scanning. So this type of genotype is going on throughout the genome at these millions of SNPs all at once. Okay, so the alleles are identified or called by the software, and some amazing how they do this. Okay, so what this looks like, here's a picture of one of the arrays, and this one has room for 12 samples. Each horizontal bar represents an array for one particular person's sample. And then the picture to the right shows the teeny little dots that are represented by the beads. And there's a small square on the second row, which is outlined. And that's what's shown in the larger part on the right hand of the picture, that just that little tiny little square is actually got literally millions, well, thousands of beads that are all being assessed at the same time. So the colors can be green, red, or yellow, depending on whether it's hybridizing to just one allele or another allele or both. So the heterozygote would be yellow because it's hybridizing to two. Okay, so in summary, where we are now, we have the person's DNA. It's been shredded and been hybridized to a lot of different oligos to determine which alleles are present at all these millions of locations. So this determines the genotype for a person for a huge number of locations in the genome. So each spot represents a particular location where variation occurs among individuals. Okay, so the next thing to think about is how we're going to use this to track down disease genes. Well, we're not going to be able to identify the disease gene directly. What we're going to be able to do is figure out what it's near to, within a few kb or so. So it gives us a good idea of the location, but does not directly tell us what the disease gene is. So most of the early studies, at least, involved genotypes, comparing the genotypes of people with the disease, those are cases, to people without the disease or controls. Now this has been extended to other types of phenotypic variation, but many studies are done by this case versus control study. Because the idea is to figure out which allele of which SNPs are correlated or associated with a particular disease. And to do this, you need apparently thousands of samples to get it right. Okay, so what's being genotyped here is just a reminder and a kind of heavy-duty statistics, which is beyond me, believe me. Just in this case, comparing diabetes people with normal people. Okay, so you can see for this first highlighted section of SNP, the people with diabetes might have a G or might have a T at that location. People without diabetes might also have a G or a T. Now, there may be some bias in the sense that normal people have somewhat more likelihood of having a G, but that's not statistically significant in this case. Okay, for the other SNP, the next one, the people with the diabetes have mostly an A, occasionally a C, and people without the diabetes have almost always a C. So that can be assessed, that probability can be assessed, or that correlation can be assessed statistically. And what that is going to tell us is that the probability of that association occurring by chance, or to say randomly, is 1 times 10 to the minus 7, which is considered statistically very significant in this regard. And we'll get back to that idea a little bit later. Okay, so common diseases are what are considered of most interest for this, things that many people have: diabetes, extension, hypertension, heart disease, etc. Okay, what's shown here is what's called a Manhattan plot, and this is a typical way of representing the data from doing these genotyping SNP analyses from a lot of people. So it's an interesting representation where the entire genome is represented on the x-axis, chromosome by chromosome, with chromosome 1 at the left going through chromosome 2, 3, 4, etc., each with different colors. So what the colors are measures of the correlation of particular SNP genotypes with that particular disease, in which case we're talking about myocardial infarction and also coronary artery disease. So what we're showing here is the probability of association of all these millions of SNPs with early heart attack or coronary artery disease. And you can see that most of those points lie very close to the 0 on the x-axis, which is to say that the probability of the association of those SNPs with the disease is very low. But here you get to the chromosome 9, the P arm. You can see there's an enormous number of points which are well above the x-axis on the y, and that represents SNPs where there is a high correlation with the disease. Now this is a log scale, so this is a log base 10 so each tick mark is actually a 100-fold increase in correlation between that disease and that particular SNP region. Okay, so the y-axis is a measure associated with the probability that the observed correlation is random due to chance. So something like this is a huge signal that there's something important going on in that region of the genome. So this is what I just said. Each little dot represents a correlation between a particular SNP genotype and the disease. Okay, so most of the SNPs are not associated with the disease, but there's a very strong association with this particular location on the P arm of chromosome 9. It does not tell us what the gene is, or even specifically where it is, or its role. It's simply a correlation. So here's an early study of several different diseases where signals, this is coronary artery disease, the one we just talked about, Crohn's disease, hypertension, rheumatoid arthritis, type 1 and type 2 diabetes. So this is typical results of these kinds of studies. Okay, so there have actually been over 3,000 studies that have been done by now. So this is a massive undertaking by many, many labs. Okay, so the idea here is that closely linked sequences stay together for many generations. And this is the idea of haplotypes. So the average haplotype in human is between 5 and 22 kilobases, but many of them are actually longer than that. So we luck out in this, in that this haplotype organization of our genome enables us to genotype a relatively small number of SNPs to get a good correlation with any disease. Now, relatively small means 300,000 to 1 million, so it's not exactly small. But if we just had a random assortment of all these SNPs, it would be impossible. We wouldn't be able to do it. Okay, so looking at our picture of our haplotypes again, we saw this before, SNP1, SNP2, SNP3, SNP4. Now these are actually, this is just a representation. These SNPs are actually spread apart from each other by thousands of base pairs. But the idea here is if all these SNPs associated, sorted randomly by independent assortment, there would be 64 different genotypes possible for these 6 SNPs. We'd have 2 for SNP1, times 2 for SNP2, times 2 for SNP3, etc., so we'd have 64 different genotypes. But because of the low recombination or non-random recombination in our genome, most of the chromosomes that exist can be represented by these 4 haplotypes that are shown. So SNP1 and 2, etc., do not assort independently. They stay together through the generations to form this block of nucleotides where these particular SNPs are still associated with each other even after many generations. Okay, so there are not 64 different genotypes here in people. There's mostly just 4, but a few others, a few minor haplotypes as well. Okay, so this represents a haplotype block where there's been no recombination in this region for many generations. Okay, this is called linkage disequilibrium, which is kind of a weird term. What it means is that there's not random assortment of these alleles. They have stayed together through the generations. Okay, so here it looks like, again, we can see them. Those are the haplotypes that are most prevalent. And the issue comes to how do we then genotype the haplotype in a way that we can tell what part of the genome it comes from. Okay, so the idea of tag SNPs is important. Linkage disequilibrium says we have large blocks of DNA that are inherited as a unit, and large pieces remain intact. So we can use this fact to design SNP probes that will represent each haplotype without having to genotype every last SNP. And these are called tag SNPs. Okay, so here's just an example of what this really looks like. We've got these 5 haplotypes, which are the predominant ones. And this is to illustrate the fact that they write them together just to annoy you, but they're actually spread over tens of kilobases throughout the genome. They're really not sitting right next to each other. So this is a region where there are 11 SNPs in fact, but these only occur mostly in about 5 haplotypes. Okay, so ignore the first part of this slide, which is enormously confusing. But the idea here is we've got 4 haplotypes, and we want to choose SNPs that we can genotype across this region, which includes actually 20 different SNPs, which is a relatively large region of the genome. So what's shown here is the SNPs actually, not the genomic region. But the important idea here is the highlighted ones. You can have either an A or a G. You can have a T or a C at the second one, or you can have a C or a G at the third one. And so all we're going to do is genotype those 3 SNPs out of these 20, which represents tens of kilobases across the genome, in order to basically identify which haplotype that is. This cuts down on the work considerably, obviously. So you only need to genotype those 3 to tell which haplotype you have, and that's the important point. So if you put these together, if it's haplotype 1, your tag SNPs are going to detect an AT and a C. If it's 2, it's going to be AC and G. If it's 3, it's GT and C. If it's 4, it's ACC. So this allows you to genotype that whole region based on only 3 SNPs rather than 20. Okay, important take-home from this. We're looking for the haplotype that has a disease gene on it. All right, important idea is that the mutation causing the problem occurred on an ancestral chromosome within one of those existing haplotypes. And importantly, it's still there because of linkage disequilibrium. So what we're trying to do is identify the haplotype which is associated with the particular disease. And of course, the idea for tag SNPs is that genotyping only a few of these can represent 10 kb or more of the genome. So this is a happy consequence of linkage disequilibrium. We can get by with many fewer probes and any SNPs to genotype. Okay, here's a slightly irrelevant example to show how this kind of works. This example uses STRs to map a disease, but the idea is the same. Okay, this is a family where they have autosomal recessive deafness in the family, and both parents are heterozygous for the mutation. And this is going to appear on the blue chromosome. So we don't know what the gene is, but we know of some STR loci that are nearby. And we can designate those alleles and track the gene based on what STR alleles the person is carrying. So yes, our STR loci can be described as having haplotypes as well. So in this family, the dad he's heterozygous for this deafness gene, and it occurs on this blue chromosome with STR haplotypes 32143244, representing those STR loci that are scattered along that region of the chromosome. So that's the haplotype where the deafness gene is located. And you can see when they have children, their deaf child is homozygous for all those STR loci, but they also have the deafness. So it's a correlation, but it does allow you to tag the genomic region and identify where the deafness gene is located. Okay, so the idea is the same. This particular mutation that caused the deafness occurred on one chromosome in one person at one time, and it's been passed down from generation to generation, along with the adjacent STR loci in this example, but it's still there. So a person that inherits these particular alleles of the STR, this particular haplotype, are more likely to be deaf. Okay, so some of the children will inherit the deafness gene, but it's important to recognize with this kind of analysis that not everybody that gets those particular alleles is going to be deaf. It's just that that particular deafness gene is in the location of those STR loci. So again, it's not a cause, it's just a correlation. But it's a good way to track it in the family if you know especially that that family has that gene segregating. Okay, so this is our haplotype, and it's associated with the deafness gene. It's still associated even though it's been passed down through the generations. Now, the recombination will eventually separate the further STRs from the deafness gene, but that's, for now, it's a useful predictor. Okay, so we could use these SCR alleles to predict in advance that the child would be deaf knowing that the parents are heterozygous for that particular mutation. Right? Recombination in later generations would let us narrow down this mapping more precisely, ideally. Okay, so the same is true over evolutionary time for SNPs. You will have mutations and you will have recombination eventually that will erode the ancestral arrangement, but some nearby sequence similarity is retained. So this is what helps us hunt down the disease gene. If we can figure out what it's close to, which haplotype it's associated with, we can then nail it down and then find the actual causal mutation. Right, so this is represented here, an example, where we have a red haplotype and a blue haplotype. And individual people have one or the other, or they could have both, they could be heterozygous. But the idea is that one of these haplotypes, some of the examples of this haplotype carry a mutation that is relevant to a particular disease. So not all the red people will get the disease or all the blue people, but there will be a correlation between the red haplotype, for example, and the disease itself. All right. So the idea is a long time ago a mutation occurred. It was in the red haplotype, and importantly, it's still there. So people with the red haplotype are increased risk for the disease, even though many don't have that particular mutation. It's a matter of correlation. But the many SNPs that characterize the red haplotype will help us figure out where the disease gene is by locating the red haplotype and associated with it to have some predictive value with this disease. Okay, so the analysis looks for a higher proportion of particular SNP haplotypes across the genome in people with the disease versus people without it. So looking at all those millions of differences along the chromosome is going to try to test statistically which of those haplotype associations is actually meaningful. Okay, again, this is done with heavy-duty statistics, that it's a correlation only, but you can get some pretty amazing correlations. Okay, so there's good stuff and bad stuff. Some genes and mutations have been identified as causing the various diseases. They're not like Mendelian diseases, and it turns out that many diseases or phenotypes are caused by small variations in many genes. So you can actually have many locations in the genes that are associated with that phenotype, not a single one like you would see with a Mendelian type of disease. So you can get maybe too many hits sometimes, which make it hard to figure out what exactly is the important one. The other thing is that identifying a disease region might only increase somebody's chances of getting the disease by a fairly small amount. It's not necessary that if you have an allele that is predictive of a high frequency of diabetes that you're actually going to get it. So it might not be as predictive as you might like to be used clinically. Okay, so here's an example of a really important region, it turns out, for being actually predictive of coronary artery disease. That location on the P arm of chromosome 9 that I showed you a long time ago. This is blown up to show just that particular region of the chromosome, not the whole chromosome. So each of those little dots above, on the y-axis represent a SNP, which is associated with coronary artery disease. Now, there are only two protein coding regions in this particular vicinity, and both encode cyclodependent kinases. And it turns out that neither of those protein coding genes has anything whatever to do with a coronary artery disease. So we can say that this particular region has been analyzed and this association has been repeated many, many times, and it's definitely real. It's not some weird statistical artifact of the early study. And the fact of it is that there are only two protein coding regions, and they turn out are not involved with arteries. But there is a long non-coding RNA encoded there. And it turns out that this long non-coding RNA is actually regulating many, many genes involved in vascular smooth muscle cells. And that this is a real correlation because these vascular smooth muscle cells are key in the development of coronary artery disease. But this is kind of disappointing to people that want one particular mutation in one particular protein that's going to lead to a development of a drug target. But it's scientifically very interesting because it tells you a lot about, or will eventually tell us a lot about, how the development of these vascular smooth muscle cells works and the various genes that are involved in those particular cells. So these cells are really deranged in people carrying the double risk allele, but it doesn't really help a lot in terms of preventing or treating their coronary artery disease. So here's what it looks like. The region, the non-coding risk interval, is 58 kilobases long, and it does encode this long non-coding RNA. And on the top is the list. The SNPs have names that are all called RS something. Those are the SNPs that are strongly correlated with coronary artery disease, the risk alleles of those particular genes. But identifying the RNA is really only the first step in trying to understand the role of this region and the basic biology, really, of this particular coronary artery disease. So obviously, much follow-up is required to figure out what's going on. And some companies are actually abandoning the whole genome approach and just sequencing what's called the whole exome, which is to say sequencing the equivalent, well, the cDNA equivalent of RNA, just to focus on protein coding regions, rather than regions of the genome which don't encode proteins but are in fact regulatory. So they're interested in direct drug discovery by identifying particular targets for drugs, which is a good thing. On the other hand, the not finding important regulatory regions in non-coding regions of the genome helps us really understand the biology an awful lot better.